So my report is about chapter 13, which is microbe-human interaction. So human host is a state of dynamic equilibrium with microorganism. So in the healthy individual, this balance is maintained as a peaceful coexistence and a lack of disease. In human hosts, we have colonization, infection, disease, and a continuum. The body surfaces are constantly exposed to microbes. So when we say infection, pathogenic microorganisms penetrate the host defenses and enter the tissues and multiply. So this is how we get the infection. So in disease, the pathologic state that results when something damage or disrupt tissue and organs. Then the infectious disease is the disruption of a tissue or organ caused by microbes or their products. So residence biota. So residence biota are the human's inhabitant. So a large and diverse collection of microbes living on and in the body is called normal biota, or other term is resident, indigenous biota, and some biologists prefer normal flora. So a metagenomics being used to identify the microbial profile inside and on humans. Okay, so the normal residents include an array of bacteria, fungi, protozoa, and viruses. These organisms have a profound effect on human biology. So next is the Human Microbiome Projects, or HMP. So HMP is being conducted at laboratories all over the country and funded by natural institutions of health. The first aim is to collect genetic sequence in the gut, respiratory tract, skin, and other body sites to determine which microbes are there. Second is to determine what role these normal biota play in health and disease. So in June 2012, members of the HMP consortium published several papers containing results about residency microbes they found in their studies of 242 healthy US volunteers. Okay, from samples were collected from 15 body sites in men and 18 body sites in women. In all, they analyzed 5,000 samples. The site included the mouth, nose, skin, lower intestine, and 3 vaginal sites. This kind of analysis just become possible due to the knowledge gained from the Human Genome Project and the decrease in cost and time involved in doing whole genome sequencing. So, human cell containing about 23,000 protein encoding genes. The microbes that inhibit has contain about 8 million, meaning their genetic contribution is about 360 times greater than our own. Most of the proteins produced by these genes are enzymes that help us digest our food and metabolize all kinds of substances that we could not otherwise use. And actually, we have a lot of microbes in place we used to think were sterile. The most striking example is the lungs. They were previously thought to be sterile, but actually containing 2,000 microbes per square centiliter. And all healthy people seems to also harbor potentially dangerous pathogens in low numbers. This is a tribute to the normal biota that rarely cause diseases. Their presence keep the pathogen in check. Acquiring resident biota. So the human body offers a seemingly endless variety of environmental niches with wide variation of temperature, pH levels, nutrients, and oxygen tension, occurring from one area to another. Because the body provides such a range of habitats, it should not be surprising that the body supports a wide range of microbes. So, acquiring resident biota, site that harbor a known normal biota is the skin and its contiguous mucous membrane, upper respiratory tract, gastrointestinal tract, outer opening of urethra, external genitalia, vagina, internal ileal canal, external eye. So in this table, 13.2 is the sterile anatomical site and fluids thought to be sterile. So our internal tissues and organs first. So the first is the heart and circulatory system, liver, kidneys and bladder, lungs, brain and spinal cord, muscles, bones, ovaries or testes, glands, and sinuses, middle and inner ear and internal eye, and fluid within an organ or tissue. First is the blood, urine in kidney, cerebrospinal fluid, saliva prior to entering the oral cavity, semen prior to entering the urethra, amniotic fluid surrounding the embryo and fetus. And for the last topic is about the initial colonization of the newborn. The uterus and its content used to be considered sterile during embryonic and fetal development. A growing number of doctors and scientists believe that fetuses are seeded with normal microbiota in utero, and these microbes are important for healthy full-term pregnancies and healthy newborns. At any rate, we know that exposure occurs during the birth process itself. When the baby becomes colonized with the mother's vaginal biota, babies born with cesarean section typically are colonized by adult skin biota within 8 to 12 hours after delivery. The vaginally delivered newborn typically has been colonized by bacteria such as Lactobacillus Prevotella and Schnitzia, acquired primary from birth canal. Um, Data from the Human Microbiome Project revealed that the microbial composition of the vagina changes significantly in pregnant women. On a Lactobacillus species that digests milk begins to populate the vagina. Immediately prior to delivery, additional bacterial species colonize the birth canal. So the scientists suggested that the lactobacilli provide the newborn baby with the enzyme necessary to digest milk, and that the later colonizer are better equipped to protect a newborn baby from skin disorder and other condition. And that's all for my report. Thank you.